Today we are talking about the fate of a cell. A fate is the outcome of the developmental decision as to what kind of cell it is. This usually happens by the time a neuron has extended its axons and dendrites. Cell fate can be determined by asymmetric segregation of fate determinants or cell-to-cell interactions. Cell fate determination in Drosophila sensory organ provides an excellent example of this interplay. Each external sensory organ consists of a socket cell, a hair cell, a sheath cell, and a sensory neuron. These cells derive from a common sensory organ known as the SOP through asymmetric cell division. NUM is a protein product that normally acts to determine cell fate within the SOP lineage. In a mild case of a NUM mutant, sensory neurons and sheath cells were missing and were replaced by additional socket and hair cells. In extreme cases, all cells became socket cells. When NUM was overexpressed, extrasensory neuron and sheath cells were produced at the expense of socket and hair cells. How does this happen? Well, the distribution of NUM is critical. Before the SOP divides, NUM localizes to one side of the SOP and is preferentially inherited by one of the two daughter cells. This occurs in further divisions as well, giving rise to cells with different fates within the SOP lineage. So how does asymmetric segregation of NUM confer distinct fates on different daughter cells? Well, the main action of NUM is to inhibit the function of Notch. Notch is a transmembrane receptor implicated in diversifying cell fate during development. Notch is activated by delta, which is a transmembrane ligand from a neighboring cell. This results in intramembrane proteolytic cleavage, which releases the intracellular domain of Notch to the nucleus. In the nucleus, Notch binds to its partners called mastermind and suppressor of hairless in Drosophila and activates target gene expression. Among these target genes are Notch and delta themselves. Notch transcription is upregulated and delta transcription is downregulated by Notch signaling. This is an example of a feedback loop and accounts for how Notch delta signaling determines cell fate through lateral inhibition. Note that this is not the same lateral inhibition that we have learned in the visual system. Suppose that two neighboring cells labeled A and B initially express the same amount of Notch and delta. If Notch activity is inhibited in cell B by intrinsic factors such as NUM or extrinsic factors, delta expression is no longer inhibited. As the expression of delta in cell B increases, since it is no longer inhibited, Notch signaling in cell A increases, while delta expression in A decreases, so that cell B receives less ligand for its own Notch. Thus, the Notch delta signaling amplifies an initial small difference between neighboring cells, leading to activation of Notch signaling in one cell and suppression of it in the other. The result of this process leads to differential expression of Notch target genes and eventually distinct fates among neighboring cells. NUM biases the Notch delta signaling to create stereotype decisions.